Hi guys. Today I'll be taking you through Lab 4 electroencephalography demonstration. My name is Dr. Matt Miller and I'm an associate professor here in the School of Kinesiology, and I'm the director of the Performance and Exercise Psychophysiology Lab, or the PEP Lab. A lot of times I teach undergraduate motor learning and performance classes as well. And today I'll be integrating both my research and my teaching by telling you about EEG, which is short for electroencephalography, and which is the main technique that we use in our laboratory. Alright, so first I'm going to tell you how EEG works. And then I'll begin talking about what it is good for and what it is bad for and why. So EEG works by placing electrodes on the skull. So here we have a mannequin with a cap, and in each place around the cap here, you can see that there are little places where an electrode can go in. So a little metal tip that is connected to a wire, and those wires will ultimately go into an amplifier to make the signal that we detect from the brain larger, and that gets recorded on a computer. So how this looks then is we have the skull here, and then we have our places for the electrodes on the skull, and we have wires that run out of them. And those all go to an amplifier, which I'll abbreviate AMP, and then that goes to recording computer, which I'll abbreviate REC-COM. So one important thing to realize is that these electrodes that are on the skull here, fitted into the cap, they are recording activity in real time from our brain. So they are recording activity from millions of neurons in the brain that are firing. The neurons in the brain fire by creating something called postsynaptic potentials. Specifically, imagine neuron A and neuron B. If neuron A wants to communicate with neuron B, it is going to change the electrical potential of neuron B by sending neurotransmitters to neuron B, which will go from the axon, or the outreach, the end of neuron A, and they'll go across a synapse, kind of a little open space, where they'll land on the dendrites, which look like tree branches coming out of neuron B, and that'll cause a change in the voltage of neuron B. So imagine that happening with millions of neurons. You're going to get a summation of changes in electrical potential. And that summation of the millions of neurons change in electrical potential is what is actually recorded by the EEG. Now specifically, the electrode underneath each one is going to be millions of neurons. So even though a really nice thing about EEG is that it can record these changes in voltage that are happening at the millisecond they occur, EEG is very bad at telling us where exactly in the brain those changes are occurring. So remember, we have electrodes here, and under each of these electrodes are millions of neurons. So it's not going to tell us exactly which ones, or even close to being exactly which ones, are actually changing. It's just going to tell us, well, we think that neurons in the frontal part, or we think that neurons in the posterior part are changing their potentials. But it doesn't do a good job of telling us exactly which ones. And not only that, there are another couple of problems. One is only neurons that are pretty close to the skull, that are pretty close to the outside of the brain, in an area called the cerebral cortex. So the brain, showing you a side view here, has an outer layer, and that outer layer is called the cerebral cortex. And only neurons in the cerebral cortex have their electrical potentials reach the skull. So that means that not only is EEG not good for figuring out exactly where in the cerebral cortex the neurons are changing their voltage, but we're also limited in that we can't look at structures that are deeper in the brain that might be of interest to us. Finally, even if you're recording electrical activity from an area of the brain, like let's say an electrode right over here around the front, that doesn't mean that it's only getting electrical signals from neurons in the cerebral cortex right underneath it. It could also get electrical activity from neurons that are somewhere else, but are oriented or pointing toward that area. Now, neurons like those aren't going to make too big of a contribution to what we're recording at the frontal region compared to neurons that are actually right underneath it, but they still have some influence. And finally, the last thing is, all of this electrical activity, when it comes from the brain, it hits the skull and it splatters. Imagine holding a piece of cardboard in front of you and standing behind it with a hose. You could tell where I am, if I'm on the right side or the left side or in the middle of the cardboard, but you couldn't tell exactly where the hose is. It's the same idea with the electrical activity from the neurons. When they hit the skull, they splatter and spread. So all this goes to say that EEG is very bad for telling us exactly where in the brain changes in neural activity are occurring. But to end on a positive note, remember that EEG is recording the moment that these changes in brain activity happen. So what we call this then is we say EEG has poor spatial resolution because it doesn't tell us where in space things are happening, but it has good temporal, which means time, good temporal resolution because it tells us exactly when neural activity changes in the brain are happening.